Good afternoon, everyone, and thank you for joining the School of Health Colloquium, Past, Present, and Future Therapeutics for Multiple Sclerosis with Peter Darlington. Peter is an associate professor in the Department of Health Kinesiology and Applied Physiology here at Concordia. Before we begin, I just want to ask everyone, if you have any questions, please enter them in the Q&A feature at the bottom of your screen, and we'll take questions at the end of the talk. Thank you for joining, and now for Peter.
Well, hi, everyone. I wanted to thank the School of Health for inviting me to do this colloquium, and also Wendy Coonan for all the hard work that goes behind organizing something like this. It's such a pleasure to be here, even on Zoom. I see there's 23 participants, and it's being recorded for posterity. So what I'd like to do today is to introduce multiple sclerosis and talk about some of the fairly recent past, present, and then some future ideas for therapeutic development. We'll focus on the immunological components, but as you'll learn, there's a lot more to this unfortunate disease that has to be treated. The first slide is to introduce myself. So I'm an associate professor at Concordia University, Department of Health Kinesiology and Applied Physiology, also Department of Biology. So hello. Multiple sclerosis is a chronic disease. It will last almost the entire lifetime of a person. As an inflammatory disease, there's also a degeneration of the central nervous system. The brain and spinal cord will have relatively randomized lesional points where the brain stops functioning due to loss of myelin or loss of axons over time. It's the most frequent neurological disease in younger people. With aging, dementia and stroke take over, but for young people, this is the most frequent. And unfortunately, it affects females at a 3-to-1 ratio. So for every one male who has multiple sclerosis, there's 3 females. And this number is even higher in some studies. Canada happens to be a very high risk zone. It may even be the highest risk in the entire world, and you can see the numbers. So we're dealing with a very serious neurological disease that affects young people that's chronic, and currently it has no actual cure. What is the cause of multiple sclerosis? I was at the MS Canada conference last week, and there's been a lot of research into what happens before the first symptoms and can we find that, and it's progressing. There are a number of genes as well as environmental factors that combine to cause a dysregulation of the immune system. This is mostly a concept map, so you can see that a number of genes, there's no one gene that causes all of the risk. It seems to be an aggregate of many genes. And there's also genetically identical twins who grow up together. One gets multiple sclerosis and the other doesn't. So we know from that that there's other factors such as Epstein-Barr virus, vitamin D, and a whole list of other factors. Today we'll be focusing on the immune system. In particular, there's a kind of immune cell called a T cell. So for whatever reason, this combination of bad luck results in T cells. Now, normally, what a T cell does is it can tell the difference between self-tissue and foreign tissue. So what that means is that the T cell can tell the difference between a virus and your own brain cells. But unfortunately, in multiple sclerosis people affected by this disease, the T cells start to attack the central nervous system. So it's this dysregulation that's led to a number of therapeutic options. I'd like to share with you a list of symptoms that people with MS are affected by. And we use this terminology now, people with MS or people affected by MS, instead of saying MS patients, which was the language we used to use in the old days. So people with MS can have some of these symptoms. They can have one, they might have one and then another, and not every person with MS has the same group of symptoms, but you see a long list here. We won't go over the whole list. Many of these are treatable with other types of therapies and drugs. You have pain and fatigue and sexual problems, bladder problems. One of the most serious aspects that affects quality of life is the progressive mobility. So the person with MS will start with a weakness in their leg, a slight limp. They'll move towards needing a cane, being in a wheelchair, and finally being bedridden. And this is really the difficult part of the disease. There's a big impact on their personal lives as well. So the divorce rates and the job loss rates are higher in people with MS as compared to the general population. So the prognosis here is not fantastic, but the good news is that over the years, they've developed a whole array of drugs and therapies that can be used to minimize the impact of the disease and improve quality of life. Now, before we get into that, I wanted to show you a simplified version of the clinical course of multiple sclerosis. And these are 3 different patterns, you might call them. And typically a person with MS will have one of these patterns. They're just general schematics because in actual fact, the presentation of the disease is quite different. So neurologists have come to the conclusion that we can have in the top of the slide is what's called primary, sorry, the Zoom is hiding my parts of my slide here. Let me hide that. Relapsing, remitting multiple sclerosis is the first thing on the list. Now, what you're looking at is a disability scale where the higher on the y-axis is the more disability. So the people with MS have what's called a relapse. Usually the first one is what brings them to the doctor. Although I saw research last week that the number of doctor visits for other reasons goes way up in the people with MS before they actually get seen for MS, which was interesting. Now, what you see then in this form is they actually go back to being almost normal, and then they have another relapse, they're normal, but at some point, the disability starts to retain. So between relapses, they're retaining disability and eventually start to progress. The second form has relapses, but then quickly moves into a progressive state. So here, like, let's say their toe was tingling, their shin is tingling, they can't feel their leg, so it's progressing over time. It might plateau, another relapse. And finally, the most difficult pattern to treat is called primary progressive multiple sclerosis, where from day one, the person starts to have a rapid accumulation of disability. Maybe it plateaus, but it will continue to escalate without the intermittent relapses. Now, one of the reasons it's important to know this for therapeutics is that it's quite well accepted that the relapses, you see these spikes in the profiles, are due to the immune system entering into the central nervous system and causing a temporary lesion. In the lesion area, the myelin is not functional, perhaps the axon is killed, and then they lose function temporarily. So what you can see then is that as the disease patterns change, you have no relapses here. So drugs that inhibit the immune system, they can control the relapses pretty well. However, they cannot slow down the progression time. So a person with effectively controlled relapses would still reach this final state without having the relapses. So that's a massive improvement in quality of life because they don't have the symptoms. Whereas immune drugs simply do not work on primary progressive patients. In last week's conference, there was a lot of research into how to handle that. So moving along then, I'd like to shift to immune therapies. We won't go over this whole graph, but we wanted to show that in 1993 was the very first immune-modulatory drug. And what they found quickly was that this drug will limit the number of relapses and also reduce severity of relapses, although it never stopped the progression. So even in well-controlled relapse people, the progression continued. And every 3 or 4 years, there's been another major drug. They have different mechanisms of action, but the common factor amongst all these drugs is that they will modulate the immune system. Talking to neurologists, I found out that when they prescribe one of these drugs, only about 30% of their patients, I could call them patients, the doctor seeing them, will respond to the drug. So 70% of patients roughly will not respond to any given drug. There are a lot of options now, so they might try a different drug. And unfortunately, over time, some patients will not respond to any drug. They lose the effect, and at that point, they're called treatment refractory, and there's very limited options at that point. So the immunological treatments by drug have been having mixed reviews, and of course, doctors would love to stop the disease in its tracks or even have a theoretical cure. which would involve some element of regenerating the brain tissue, and other scientists are working on that. So far I mentioned to you that people with MS have a chronic disease with many symptoms that can be relapsing, remitting, and it has this underlying progressive element. You notice that there wasn't any time frame on those graphs, and you might be wondering, how long do those events happen? And there's really no way to predict that. So I've heard from neurologists that some people, upon diagnosis, have only 3 years and they die from their multiple sclerosis, sadly. Other people can live for 30 or 40 years. The average life expectancy of a person with MS is about 7 or 8 years shorter than the average life expectancy. So really what we're talking about is quality of life and trying to slow down the disease at this point. So I'd like to quickly go over 3 chapters here. One is to discuss one of the most powerful and exciting new therapeutics that's available now as a treatment. It's called bone marrow transplantation. We'll then move along to some generic drugs that my laboratory here at the School of Health has been studying, and we found some with therapeutic potential. And finally, I'll briefly introduce a new project we've developed with Dr. Helfield regarding something called focused ultrasound. Now, in approximately the year 2000, the Canadian Bone Marrow Transplantation Study Group formed, led by Mark Friedman, and who's a neurologist at the University of Ottawa, and Harry Atkins, who's a hematologist. And Harry Atkins, his specialty was in chronic myelogenous leukemia. Chronic myelogenous leukemia is a state where the T cells become cancerous, and the best treatment for that is to use chemoablation or chemotherapy, followed by a bone marrow transplant. So what Mark and Harry did, with the help of the team, was to develop a adapted protocol for multiple sclerosis. The concept here was, if there's something going wrong with the immune system, then maybe we should essentially wipe out the immune system and replace it with a brand new immune system. One of the key differences between the leukemia procedure and the multiple sclerosis procedure is here we're using what's called autologous stem cells. That means they're from the actual person who will be receiving them. Of course, in leukemia, they have to find the stem cells from a close relative or a matched donor. So in the center here, we have a person with MS. The first step is what's called leukocytophoresis, where they run their blood through a machine, which will extract the stem cells. The stem cells are then processed to remove any of the residual T cells. As I mentioned earlier, the T cells are generally considered to be the bad guys in multiple sclerosis. We'll develop that idea a bit more as we go on. Next, those have to be cryopreserved in a freezer very carefully to keep them sterile and to preserve them. And then comes the big step. So the person with MS will get a potent dose of chemotherapy, which will eliminate their entire immune system and all of the stem cells in their body. At this point, they have to be in the hospital. They're in a sterile room for up to a month, and they're closely monitored for infections. They have to take antibiotics, antifungals. They're completely vulnerable at this point to infections. After about a month, their immune system comes back because the doctors will quickly, after the chemotherapy is stabilized and the person's stable, they reinfuse those stem cells and allow them to regrow into the blood. So this was a very invasive procedure. It had a 4% mortality initially. So one person died in this study. They redid the ethics approval, redid the consent, and readjusted the chemotherapy regime. And now the global number is 1% at this point for mortality. So 1% is a pretty good number. It's risky, but keep in mind that this type of procedure is only available for patients that are treatment refractory, and they're also at a very advanced stage of disease. So ethically speaking, this can only be done when there's no other viable options. So it's a high intensity treatment. The idea is to replace the old immune system with a new immune system. There's less morbidity and mortality compared to the procedures used for chronic myelogenous leukemia. And the Canadian bone marrow transplant study, I'll just summarize. There's been lots of papers written by Mark and Harry. It was very successful. So the patients in the study who got the transplant, they had no additional relapses afterwards for up to 10 years of follow-up. One person actually had a reversal of their symptoms, which is almost unheard of in any other treatment. And on balance, the symptoms were essentially plateaued or neutralized, if you will. So the data I'm showing you here is actually a meta study, because there's been dozens of similar studies all around the world. And so doing a meta-analysis of 764 patients, you see a lower disease progression. Now, one of the really exciting features of this therapy is that the person with MS does not have to take any more drugs afterwards. So this is a one-time therapy, about a month in the hospital, and all they have to do is come back for follow-ups and make sure they're not getting fungal or bacteria infections. So it's very well tolerated, and so far it seems to be a permanent, lasting effect. Okay, so that's a lot to take on board. Now, what was my role in all this? I was actually part of the team that was analyzing the immune system, and we were trying to learn what happened to the patient's immune system in more detail. Before I get into the next couple of slides, I'll just have to explain a little something about T cells. So T cells, as I mentioned, are at the center of determining between self and non-self. They also have what's called pathogenic versus suppressive types or subtypes. So T cells have different flavors. We got blueberry, we got strawberry, we got grape juice, and we got tangerine. Fantastic. And these cells have properties. Th1s, the T helper 1 cell, and the T helper 17 cell are considered to be pathogenic, whereas the T helper 2 and the T regulatory cells are considered to be suppressive in the context of autoimmune disease. There's lots of letters and numbers on this slide. The important one is interferon gamma. This is what's called a cytokine. It's kind of like a hormone of the immune system. It's made by Th1 cells. And we have IL-17A, which is another cytokine made by Th17 cells. IL-4 and IL-5 are made by Th2 cells. And I don't have much to say about Tregs, but we'll have a bit of data later. So as of the beginning of this study, the hypothesis was that the pro-inflammatory Th1 cells would be decreased in the patients after the therapy, and the Th2 cells would be increased. So that was the hypothesis. Now, the first result that was acquired, and here we have blood samples from people with MS before the therapy, hematopoietic stem cell therapy, transplantation. And we have blood samples from 12 months after the therapy. When we measured interferon gamma, it was almost exactly the same. Very small decrease, but it wasn't significant. And then more surprisingly was the Th2 cytokine was decreased, although it wasn't significant. It's going in the absolute wrong direction compared to our predictions. So this was quite surprising, the data. And around this time, the Th17 cells had been discovered and people were realizing how important they were. So with colleagues, we developed a way of measuring Th17 cells in those samples from the patients. And what we can see is on the top here is what's called a dot plot. Every dot here is a T cell, and you can tell right away they're grouping into certain areas. If it's up on this part of the graph, it's an IL-17 cell. Those are Th17s. This part of the graph is an interferon gamma cell. It's a Th1. Humans actually have a double positive cell. We call those Th117s, and then most of them are other things, which I don't want to explain right now, but there's lots of other types of T cells. So we can look at the two pre-treatment and post-treatment graphs and see that the Th17s look like they went down. 8.6% went to 3.6. The Th1s may have gone down, but it was a close call. And these certainly appeared to go down. In panel B, we analyzed the data for up to 9 participants. And what you notice is in the first panel, the Th17 cells were decreased in every participant except for this person. And that was a significant effect. The double positive cells were decreased in most people, and that was also a significant effect, whereas the Th1 cells increased in half of the people, decreased in half the people, and there was no significant change. So this more modern analysis confirms that the Th1 cells were not significantly changed by this therapy, which contradicts some of the predictions we had from the literature. And excitingly was that the Th17 cells appeared to be the best clinical correlate that we had in the blood samples. And if one could measure these two types of cells, you would get a good sense of how well the person responded to the therapy. It also makes the point that this transplantation is not just about eliminating a bad immune system, it's about replacing it with one that's more suppressive and has a better profile for the person. To understand why this was happening, we did a follow-up study looking at something called a natural killer cell. Natural killer cells, just to briefly introduce them, are primarily designed and developed to, or exist, I suppose, to kill virus-infected cells or cancer in our body. But it also turns out that natural killer cells are really good at controlling the T cells in our body. They act as a kind of regulatory cell. There's a number of publications supporting this idea, and we had the thought of testing it in the samples. So what we're looking at here is the MS patient samples from baseline, that's before the therapy. And then we followed it up with sequential blood draws. So the patient came back into the hospital at time points of roughly 3-month time frames. I didn't put all the data in this graph, but we have 3 weeks after the therapy. There's nothing in their blood samples. That was drawn when they were in the hospital in the sterile room. This is 3 months after the therapy, 12 months, 21 months. So when you look at the number of natural killer cells in the blood, the first thing you'll notice is that they come back relatively quick. By 3 months, they're at 1/3 of the baseline levels. And by 12 months, they're back to normal levels, and they seem to stabilize around 21 months. The T cells, on the other hand, they drop down to next to nothing, and they jump up to very little, and they never actually, even after 21 months, they're still not even close to what they were beforehand. They're not even at 1/3 of the original amount. So when you look at this profile, the NK cells are rebounding after the therapy really fast from those stem cell transplant, whereas the T cells seem to go a lot slower, or the NK cells are actually killing them. One piece of evidence was the ratio. So if we look at the number of NK cells to the number of T cells, in the baseline, it's only a decimal point. So there's like 1 NK cell for every 10 T cells. But afterwards, it's practically inverted. We have 3 NK cells for every 1 T cell. And it seems to settle around a 1-to-1 ratio. So for every 1 T cell, there's 1 NK cell in the blood. That is a very unusual profile. So if we take any healthy blood, NK cells only represent about 5 or 10%. Now in the paper, we have additional data that I won't show that demonstrates that NK cells can actually regulate the Th17 cells like directly, which provides a novel mechanism of action by which the stem cell therapy appears to exert its effects. And just to remind you that the people in this study did extremely well and had no more new relapses after the therapy. So it was very exciting that this procedure at the time was part of a clinical trial, so it can only be enrolled in for specific reasons. Nowadays, I just heard from Mark at the conference that it's been approved as an option now. So MS patients can now elect, if neurologists and hematologists agree, to go through this procedure as an actual treatment option. And there's such similar programs around the world, as I mentioned, that are very promising. And what we learned was that the Th17 cells were a better clinical correlate because those were the ones that were suppressed in the samples as compared to the Th1s or the Th2s. And finally, we have novel evidence that the natural killer cell might be a way of actually getting a little bit faster to the whole thing. Is imagine you can have some kind of natural killer cell therapy that would be even more effective. So that's an exciting discovery. Now, with all that in mind, the stem cell transplantation will probably always be reserved for patients that are, or people with MS that are refractory to drugs or who are simply at an advanced stage of diseases. I know Dr. Friedman always said that he'd love to be able to treat patients a lot earlier because maybe you could slow down the progression. One of the limitations of the stem cell procedure is that it's a very expensive procedure that requires a dedicated facility. And so when you think about whether every country in the world can do this, it's difficult. A center really has to commit to having the stem cell set up. It has to be sterile. It has to be good laboratory practice standards, etc. So there's also a need to discover new drugs, and especially generic drugs. And one of the speakers at the conference last week was talking about various generic drugs and the fact that when a drug is off its patent, there's a couple of advantages. It's a lot cheaper, for one thing. It's more available. And the whole risk profile of a generic drug is really well known. In case you're not aware, for a new drug to get on the market and be prescribed, it takes literally billions of dollars to get that drug from discovery phase all the way through to the prescription pad of the doctor. So that's really the advantage to looking at generic drugs is as a way of having a more accessible remedy and give neurologists more options with a drug that already has a recognized risk benefit profile. Having said that, now we don't know if those drugs are even useful. So a number of years ago, my lab developed a system to study generic drugs and determine if they have any therapeutic potential based on their ability to modulate the immune system. And we focused a lot on Th17s because that was the best clinical correlate from the bone marrow trial. And the Th1s, Th2s, and even the Tregs were of interest to study more. So from here on in, just to clarify, is the studies were done on healthy human participants. We had institutional approval and informed consent. We used the blood sample for the experiment. The person did not take the drug at all. There was no intervention. They donated blood. Once they were stabilized, they were allowed to leave. And that's been ongoing. So what we're able to do then is to take this blood fraction, which is called peripheral blood mononuclear cell. That's actually a mixture of T cell, B cell, and K cell monocyte. And we can also then obtain an even more purified type of T cell. Memory T cell is actually the one that carries the chronic inflammatory properties, keeping in mind that this is healthy participants, so it's not quite the same as doing the experiments on multiple sclerosis samples. We measured cytokines by enzyme-linked assay. The cellular analysis was by flow cytometry, and where applicable, the genes were sequenced at the McGill Genome Center. The theme of the laboratory, the main project that we have, I should say, is to study adrenergic and cholinergic drugs. Now, these are typically used for other indications such as asthma, hypertension, or cardiovascular disorders. And there is some scattered evidence that T cells respond to these drugs, but the details are unknown, and the therapeutic potential is just not clear at this point. So our overall goal was to determine if some of these drugs had any therapeutic potential, and if so, could they be further explored in animal studies or even on patient samples. The end game, of course, would be to do an actual clinical study if any of these things start to pan out. So the first project we took on was based on literature. We read about a drug called terbutaline. This is a beta adrenergic receptor drug. It's typically used to treat asthma. And we embarked on a project here. And the first result we got was a bit surprising. So participant A, this is a sample from a healthy participant, blood sample, was tested. And we got a surprising kind of opposite to what we were hoping type of response, where the IL-17 went up substantially in their sample. So this was not a desirable outcome. We had activated the cells. You have to activate T cells, otherwise they don't do anything. They just sit there in the petri dish looking back at you and wanting more glucose, but otherwise they don't do a whole lot. So you can see when you activate them, you get IL-17. When you activate in the presence of terbutaline, this is all in vitro in the dish, you get almost double the amounts. We also retested the sample on a new blood draw. So we waited about a month, we drew blood again, did the whole procedure again, and you get the same result. Although the levels are different, it still goes up. So this did not bode well for the whole concept. And rather frustrating is we also had some people where it seemed to go down slightly, which seemed to be a better profile. So in this graph, we determined that the different responses in people were due to a genetic polymorphism. That's a genetic code change that people are born with in the actual beta adrenergic receptor. So 6 people in our study here had a genetic identity of being arginine homozygous. These 21 people were arginine heterozygous, and these remaining 12 people were glycine homozygous. Now, what does this mean? These are actual amino acid components of the receptor protein. So the receptor protein has a fundamental difference between these people. And there's 2 ARGs because, of course, we have a chromosome from mom and a chromosome from dad. So that's how that's inherited. If we focus first on the black and white bars, we have activated cells, and then we have activated cells plus terbutaline. People who are homozygous for ARG in their receptor had no response to the drug, and some people actually went down a little bit, but statistically there was no change. Whereas people who had at least 1 or 2 copies of the glycine gene had a significant increase. Now, if we think about the applicability of terbutaline for autoimmune disease, it looks pretty much like a dead end because you would never want to give an MS patient anything that would raise IL-17 levels. Theoretically, that's bad. And even in these people, it didn't seem to do anything. And if we count the numbers, it means that 6 people had no effect of terbutaline, but 33 total people have the boosting effect. Now, does it work the same way in MS patients? I'd love to know. And, that's a nice future direction to get blood samples from an MS patient. Now, I'm going to explain a bit more about why terbutaline does this in the next slide. And then in the subsequent slide, I'll talk about another drug called nebivolol, which we had pivoted. We said, well, if terbutaline is not going to work, what about a similar drug called nebivolol? And spoiler alert, you can tell right away the gray bar for nebivolol drug is actually working quite nice. So no matter what the genetic code, there's about a 50% suppression in IL-17. So that, in a way, is the most exciting thing, is that to us is nebivolol. A total surprise that it worked this way, but keep in mind that also resembles a lot like what we saw in the bone marrow transplant trial. If you think back to that graph, you see like almost a 50% drop in the IL-17. Now, the story on terbutaline, the last slide on terbutaline is, I'm not going to get into all the chemicals here, but what we did was we added the terbutaline and we added a number of inhibitor drugs to figure out what it was doing. And to make a long story short, if we look at the diagram, is what we discovered in this paper was that the terbutaline will activate a pathway that then increases the amount of IL-17. The very same pathway will decrease the interferon gamma. So with that mechanism in place, I think we've reached a point where for a therapeutic, this drug is not very interesting. For basic immunology, this is fascinating because it suggests that the adrenergic receptor system might be able to boost IL-17 levels. So there's always a fundamental side to these discoveries. And the work was done by Catalina Carvajal-Gonsi and also more recently Mary Hajiagi, who completed the study revisions. The reviewers had a lot of comments about what we had done, and now it's published. So let's move along to nebivolol because it's a better candidate. So again, I'll just gloss over this, is we had reasoned that this pathway was bad, so why not use nebivolol? I don't know if anybody watches Seinfeld. I'm going to drop a Seinfeld reference here right in the middle of my MS talk, but there was something called the Costanza theory. So Costanza was a character in the show and he always made the wrong decisions. And so he at some point said, if I always make the wrong decision, I should just do the opposite. And then that's the right decision. So in the show, of course, everything goes awry. So we used the Costanza theory here. Actually, it was Catalina. And she said, if this drug is boosting IL-17, then this drug, which is considered to be what's called an inverse agonist, it must decrease IL-17. And of course, I showed you the graph already, and you saw it. So nebivolol, indeed, it was a very powerful suppressor of IL-17, and we've been able to reproduce that in our more recent studies. And in this study, we further explored nebivolol. We have a larger sample size. This is done on memory T cells instead of the PBMCs. And also, we've got 3 different cytokines measured. And this is all work by Mary Hajiagi and Fatime Holizadeh, who has also collaborated on this. And I'll show you her data too in a second. So right off the bat, IL-17 goes down. Interferon also goes down, and the IL-4 goes up significantly. And this is on 11 participant samples. These are healthy human participants. So the exciting thing is this profile matches exactly what we would want to do for an autoimmune patient. You get less of the pro-inflammatory cytokines, and you get more of the anti-inflammatory cytokine. And all the drugs we've looked at, this seems to have the best overall profile if you want to look at the context of autoimmune disease. Just sort of quickly covering the next few slides. The reviewers of our grants and also our papers kept saying, you study the adrenergic system, but what about the cholinergic system? It had always been something on the back of my mind. And when Fatime came into the lab, I asked her to tackle this project. She's done a wonderful job on a brand new project that is yielding exciting results. This is all novel stuff. The immunologists have never done these experiments after all the years. And let's just look then. We have what's called a muscarinic agonist. Muscarinic receptors are part of the parasympathetic nervous system. T cells have them. And what you notice is that this drug, which activates the muscarinic receptor, increases IL-17, increases interferon gamma, and decreases IL-4. Therapeutically, that's exactly the opposite of what I just showed you for nebivolol, which makes it not a very good candidate. For fundamental science, it's exciting because you got one thing that has a pro-inflammatory profile, the other has an anti-inflammatory profile. Is that how the body works? Is this how the immune system works? I find that basic question is just as exciting. And finally, to end on some good news with the cholinergic project is the nicotine treatments. This activates the nicotinic receptor. It starts to have the anti-inflammatory profile. Nicotine decreased IL-17, decreased interferon gamma, and the IL-4 data we're still working on. So nicotine seems to have similar promising profile as nebivolol. Nicotine, by the way, is not a prescription drug. That's the one that's in cigarettes and present in supplements. So there might be some benefits there in terms of applying it down the road. So in the last 5 minutes, I'd like to shift gears again and just to look at the big picture at this point. There's already about 25 immune modulatory drugs in the market. And as I was trying to argue is that generic drugs might be more applicable to countries that cannot afford the new drugs. They're cheaper and perhaps they have unique effects like we saw here. But at the end of the day, how do we stop the progression of multiple sclerosis? One way is to help regenerate the brain. And there's a lot of labs working on that now. They're figuring out ways to make new myelin, to make new neurons. It's really exciting. And another way is to see if we can get the immune modulatory drugs inside of the central nervous system. So just so you know, when a person takes one of these drugs, it circulates in their blood, their lymph, reaches peripheral tissues, but most of those drugs on the list, they don't go into the brain. And if we think back to the beginning of the talk, that's where the so-called action is happening. The pro-inflammatory effects in the central nervous system are the ones that are probably causing some of that progression and incurable disease. So being able to put a drug into the central nervous system in a very controlled fashion would be an exciting development. So let's shift then to chapter 3 here is focused ultrasound. Focused ultrasound, you know ultrasound as the imaging technology used to look at muscles and tendons, but did you know that those focused ultrasound beams can also be used to essentially destroy solid tumor tissue? And there's a number of clinical trials right now where doctors are using magnetic resonance imaging to guide the focused ultrasound beam and essentially zap down tumors. It's a very precise way of doing it because the energy only increases where the beam focuses itself. It can go through all the bone and skin and tendons and get it right into the tumor. It's also been used in a mouse study for dementia, where they added focused ultrasound with something called microbubbles, and that facilitated the entry of a drug into the brain, and the mouse improved dramatically its dementia symptoms compared to a mouse who only took the drug normally. So as you can see, this idea of targeted delivery of drugs to the central nervous system has a lot of potential, but to date, there's not a single study in the area of multiple sclerosis. So we decided with Dr. Hellfield, who's in the physics and biology department, and he's an expert on focused ultrasound with microbubbles, to plan out a project. So there's no new data yet, but I'll show you a few things that he had. This is data from Dr. Hellfield's lab. And the first thing you see is microbubbles. These are very small bubbles. And when you apply ultrasound, the pulsations will make the bubbles shrink and grow. And why do you do that? Because if you look on the panel B, this is the in vitro blood-brain barrier. We won't go over all the details of this slide, but just focus on this area here. So it opens and then it closes, all in the course of only 30 minutes. So importantly is this ultrasound microbubble combination can open the barrier temporarily, so it's not a permanent destruction. Now imagine that a drug can then enter through the barrier and get into the central nervous system and then quickly heal itself. And considering the level of precision you might be able to maintain, this is pretty exciting. So the proposal we developed was using his apparatus. We will focus the ultrasound beam into the flow chamber. The injection port receives the bubbles. We have brain endothelial cells growing in a membrane, and that creates a top and a bottom. So if I show the next slide, we're planning on adding a therapeutic. I didn't talk about antibodies, but these are therapeutic antibodies that are currently prescribed to people with MS. And what we want to figure out is whether the focused ultrasound beam and the microbubbles, does it destroy the antibody or does it allow it to continue doing its job? And perhaps more importantly, does it actually cross the barrier and reach its target cell, in this case, B cell? And we chose this as a very common therapeutic that's prescribed. And secondly, this is more experimental, but can focused ultrasound allow a regulatory T cell to cross the barrier to stay alive and also keep its regulatory function? We don't know the answer yet. And there's lots of parameters that we're going to test to see what's the optimal way of delivering it. If it works, it's exciting, and we'll probably look towards moving at animal studies. And the theory one day is that a person with MS can come in and have like a 10-minute procedure where the doctor will focus the drug delivery into the central nervous system safely. Central nervous system heals itself up, and then they hopefully have a better clinical outcome. That's the speculation at this point. It's nowhere near being ready for clinical use. So to summarize, I showed you that stem cell therapies are very effective at stabilizing disease progression. They don't need any ongoing drugs. However, it's something that's very technically complex, expensive, and probably won't be available to all patients until they reach a later stage of disease. So the second part was showing that generic drugs could potentially be repurposed, which opens the door for other treatment options, perhaps in countries that can't have access, or people that can't afford. Some countries don't have healthcare, so there's always a benefit to generic drugs. And finally, this new idea about focused ultrasound might be a way of helping cell-based therapies, antibody or drug-based therapies from getting directly into the central nervous system. There's obviously risk involved with that approach, and the research will have to assess this before it's a reality. And let's not forget that fundamental science always leads to exciting discoveries and breakthroughs. And there's even more details the students have on their posters, which I hope we can hang in the School of Health soon. We'll get the new posters up and you can see all the details. Just briefly, this slide is to acknowledge all the funding sources for the Canadian Bone Marrow Study Group. And I mentioned some of the key figures. Amit Bar-Or was leading the immunology studies at the time, the General Hospital. In my particular lab in the School of Health, we have our 2 PhD candidates, Department of Biology, doing the research in the School of Health. That's Fatime Olizadeh and Mary Hajiaji. We have our 2 master's students getting close to graduating now. We have Maria and Amanda and a number of excellent undergrads. And thank you very much. That's all today. Thanks for your attention.
Wonderful. Thank you so much, Peter. So now we're going to go to some questions. We have a couple already, but just to remind all of our attendees, if you do have any questions, please enter them using the Q&A feature at the bottom of your screen. All right, so we're going to go to our first question. So Peter, can you clarify who's a candidate for AHSCT? You stated 1% mortality was acceptable as done only in severe disease, but it seems most are treated early in disease course in most trials.
Yeah, that's a good question. The original clinical trial that was done with Dr. Friedman and Dr. Atkins and the team, because of at that time, the risk was actually 10% mortality. And so that was a very high risk. And so what had to happen was, ethically speaking, the patient had to have treatment refractory disease. They had to have already been put on up to 3, and some of them were on up to 8 different conventional medications. And that meant that nothing conventional was working. And they also had to have what's called a EDSS, that's a clinical score that was quite high. So they were into the 4.5 to 5 range of EDSS score. So they're very close to going into the final phase of the disease where they're going to be severely disabled or even like catatonic in a bed. That's the final stage of MS, sadly. And so ethically speaking, that's where it was, and the patients had to consent to all this as well. They were well aware of the risks. And as I mentioned, 1 person died after the initial 10 did the study. And Harry and Mark had to recalibrate the chemotherapy doses to be less harsh. They redid the whole ethics approval and they reconsented the patients. And only 1 patient actually dropped out, even knowing the full story, full disclosure. So it does show how, I don't want to use the word desperate, but, it's a decision they're making when they realize that my life quality is, and you saw in the initial clinical course, it just progresses and degenerates. So 1% mortality is still considered to be ethically, I mean, it's obviously 0 would be fine, but this procedure, the chemotherapy has a hepatotoxicity issue that it's hard to predict. And there's always a chance of infections, but that hasn't happened so far. In chronic myelogenous leukemia, it's much higher. I'm not familiar with the exact numbers, but it's 10 or 20% or so, although that number has also come down as hematologists get better. Yeah. So that's the story on that.
Great, thank you. So we have another question. Is MS an environmental disease mostly, or is it genetic? Demographically, beyond male-female differences, are there significant ethnic differences? Do we know why it is most prevalent in Canada? And thank you for a great colloquium.
Well, great questions, actually. Nobody really knows the answer to the first one, which is what's more prevalent. The biggest genetic susceptibility is called the HLA-DR. That's part of the immune system. It only confers around a 2% risk factor, which is extremely low. So it's a lot of people have that polymorphism and they'll never get MS. And a lot of people have MS and they don't have that polymorphism. So the genetics has never been clear. The latest story is Epstein-Barr virus, and there does seem to be a good correlation. But the issue there is, maybe you don't know this, but virtually 99% of all adults have Epstein-Barr virus. It's the virus that causes mono in some people. So that's never been really figured out at this point. The question about female versus male, there's no known reason for that. They've studied estrogen extensively. And there's also an interesting observation that when a female with MS is pregnant, she will not have a relapse during the entire pregnancy. It's just been a well-accepted fact by now. And a lot of researchers are trying to figure out what kind of pregnancy-related hormones might be advantageous for adopting as a therapy. The last question, it's very interesting you asked that, because about the ethnicity at the conference last week, they had pointed that out as a major deficiency in the research. And historically, it was thought that Black people were less susceptible to multiple sclerosis. And this is the research that they were mentioning. What they found was that that research was heavily biased because in countries in Africa, a lot of them aren't able to diagnose multiple sclerosis. They don't have the imaging equipment to do so, and the clinical know-how isn't quite there. So when more modern research was done, they found that the rates of multiple sclerosis were roughly the same between Black people and non-Black people in that research. And so that's an ongoing question, though, and from what I understand, it might be roughly equal between ethnicities, but again, it has to be studied more.
Thank you. So the next question, is the patient's diet ever considered as a parameter when looking at effectiveness of a drug?
A neurologist will normally not think about that whatsoever. And there's no specific dietary recommendations for a person with MS at this point. So it's not like, say, diabetes, where there's very specific guidelines. Having a healthy, balanced diet is advantageous. It's probably an interesting research area, though, as it relates to the microbiota. That's a whole nother area of science. I guess short answer, long answer, but there's a fascinating thing where they've been able to, in mice at least, they've been able to transfer the mouse version of multiple sclerosis. They can transfer it between mice using fecal transplant. So fecal transplant moves all the microbiota from one mouse to another. And then the healthy mouse can get multiple sclerosis if the first mouse had multiple sclerosis. And in London, Ontario, they're having a clinical trial on fecal transplants for MS patients. I know it sounds kind of revolting to some people, but it's an injectable fecal matter that from a healthy person into an MS patient. But unfortunately, I haven't heard any results from that clinical trial. So whether there is a dietary connection, there probably is. The Th17 cells primarily exist around the intestines and the gut, which I didn't mention earlier. And that's been something that scientists found out in the last about 5-10 years. So that's the story on diet, as I understand it.
Thank you. So we still have a few more questions. Next question. Thank you for a very interesting lecture. Are you planning on performing studies with the mentioned generic drugs in people with MS?
To get that far, there would have to be a few other steps. First of all, a mouse experiment. There's a model called experimental autoimmune encephalomyelitis. So before going into a clinical study, there would have to be some proof of concept in the mouse in vivo. So that's one thing to consider. To do a clinical study, it's not out of the realm of possibility, and there's labs that do that. So, and when we're talking about generic drugs, the threshold for doing that is a bit lower. So you'd still have to have Health Canada approval. But in theory, let's say Nebivolol was an interesting one, that could be a clinical study. Yeah, with the right approvals, of course.
Great. So the next question, could you please talk about the exact time of transplantation in these patients according to the graphs? Is it between the relapse and remit phase, or it would be in remitting phase, for example? What is the most definitive factor to define the stage?
Let me I didn't hear one of the words there. Was it?
Could you please talk about the exact time of transplantation?
Yeah, I see. Oh, that's Nagar. Hi, Nagar. Yeah, so the patients in the study, they weren't allowed to have any. No, sorry, that's they weren't allowed to have any glucocorticoids, and they had at least 2 lesions within the last year. So they had severe MS. So when they got the therapy, they had to be in relapse. They went into the hospital, they got the therapy, and the therapy only lasts for like a week, right? And then they're in the hospital for roughly a month. And then the follow-up afterwards was, Mark published follow-up papers at 5 years, 10 years, and now there's a 15-year follow-up. So they've followed them for all this time. So I guess your question is, yeah, so it would have been between relapses. They would have been in remission during this actual study.
So the next is also from Nagar. I also want to know about the drugs which can stimulate making myelin. Can you talk a little bit more about that?
Yeah, so I wanted to know about the drugs. Yeah, there are a number of drugs now, like fingolimod, that can produce myelin. So that's been researched by Dr. Moran and others, and Jack Cantel. That's an exciting other opportunity to expand the use of drugs and try to regenerate the brain tissue.
So that seems to be the last question.
Yeah, I see the comment. That's a good comment. I appreciate this comment.
Yeah, so we did have someone who wanted to add a comment saying not all people diagnosed with MS are debilitated or catatonic, and people can and do enjoy their lives and have value even with disability when they are debilitated. So I appreciate to show the use of research. You want to show the problem with MS, but those with MS often have a different perspective. So.
Yeah, I know it's a comment. I'll respond a little. I really appreciate that. You know, I came out of an immunology background, and when we study diseases and it's bad and we have to fix it, I realize that language is not helpful and for people with the condition. So, you know, that's why in a way you could tell in the talk, I kept saying MS patients because that's how I talked about it for so many years. And then in the last 5-10 years, I realized from going to these conferences, and the great thing about MS Canada is they have people with MS are at the conference. They help organize it. They help review grants. They participate. And I heard a lot from them at the conference. And you do have to see it from the standpoint of people, and that it's not just here's a drug to fix your problem, you know. So that's something as a researcher communicating, we have to figure out how to communicate things. When we do animal research, it's even more sort of cut and dry, and that's not reflecting the human condition. When I did say catatonic, I was really referring to the very last part of the disease, not when they're diagnosed. A typical diagnosis could be somebody, well, I heard this from a neurologist, so somebody left their children in a shopping cart at the grocery store, and they came home and they didn't realize what they had forgotten. And so they were sent, luckily they got the children back and there was no problem, but that was their first relapse. And I also got the chance to work with people with MS in research laboratories that were at the McGill, and they were doing research on the condition as well as having it themselves, and that was an interesting perspective. So I do appreciate that comment, and we need to figure out ways to communicate the science and to do the right kind of research too, so that MS Canada helps steer the kinds of research that are important. And there's a lot of quality of life studies. I didn't have a chance to talk about things like they have a new robot system that can help the people coordinate their hands better. So the robot teaches their hand how to move, and then after the robot's over, their hand moves better. And there's lots of exciting things like this going on. So it's fantastic to get everybody's perspective.
Well, thank you very much, Peter. We are at the time now to come to a conclusion. So thank you very much for your talk today, taking the time to share a little bit about your research. And I hope everyone enjoyed the talk. And if anyone missed it, we will be sharing it on our website in the coming days. So have a good afternoon, everyone.
Bye, everyone. Thanks.